So good morning, everybody, and welcome back from the coffee break, and welcome back from a truly fantastic introduction to our day by Jennifer Doudna. So my name is Stephen Rees, and I lead the Discovery Biology group at AstraZeneca. And we are absolutely delighted to be supporting this series of Transforming Our Life conferences in partnership with the Royal Society. This is the first of 3 meetings that will be supported by AstraZeneca over the next 18 months or so, where we're hoping to explore novel breaking technologies and how they can transform, bring societal benefit through transforming what we do as scientists and bringing that through into the general community. When I think about what I do on a daily basis, what do I do? So I work for a drug company. What is the mission of our drug company? So AstraZeneca's purpose, corporate speak, we push the boundaries of science to deliver life-changing medicines. And for every pharma company, and I suggest many biotech companies, much of academia, the end point of our research is to deliver life-changing medicines. Discover medicines that can transform patient health. Transform the future of patients, restore their health, transform the future of their families, transform the future of their friends by bringing, curing the disease, treating the disease within those patients. And in turn also transforming their ability to contribute to society. That is what we do. That is what we do. We deliver transformative medicines to transform patient health and then in turn lead to their ability to continue to contribute to the society, contribute to their families. And we've heard Peter touched on this, Jennifer touched on this. I'm also now at the stage of my career where there are more years behind me working in pharma than there are ahead of me working in pharma. And I think this is actually the most exciting time to be working in biological sciences in my career. And if you look at some of the developments of technologies and capabilities over the last 5 years or so, they truly are transformational in terms of our understanding of biology, our understanding of our ability to stratify disease, our understanding of our ability to bring medicines to patients. And I'm sure those same transformations will apply across the agrochemical industry and across the various presentations we're going to hear about in the next session. And just to think about 2 or 3 of these transformational capabilities. The first is next generation sequencing. We all know this, but maybe it's worth just reflecting a moment. We can now sequence a genome of an organism for about £500, about £500. So that's less than a cell phone, probably less than a television. It's less than a washing machine you can have in your house. You can get your genome sequenced. Now, I don't know how many genomes need to be sequenced to understand the genetic cause of all disease, but I'm willing to bet it's not 100 million or 500 million, and I'm willing to bet that data set will be in place certainly within the next decade, maybe within the next 5 years. And then our challenge as scientists, our challenge as a society will to understand what that information means in terms of being able to deliver medicines to treat that disease. We hear a lot about cell technology, stem cell technologies, the ability to derive terminally differentiated cells of pretty much any cell type from stem cells from any one of us. In the lab today, we could take a swab of cells from the inside of the cheek of anybody in this room, and we could create stem cells which we could differentiate into cardiomyocytes or hepatocytes 3 months, 4 months' time. Truly transformational, and these are the things we've alluded to it a couple of times that undergraduate scientists are doing. Artificial intelligence. How many times in the last 6 months have you heard artificial intelligence on the radio, on the news, or wherever? But artificial intelligence is here and now in terms of what we do in terms of drug discovery, helping us to select compounds, helping us to optimize compounds, helping us to bring those forward to the clinic. And of course, the subject of today's meeting, precise genome editing, CRISPR-Cas9. 6 years ago, I joined AstraZeneca 7 years ago. I'd never heard of CRISPR-Cas9. I don't think any of our scientists in AstraZeneca had heard of CRISPR-Cas9. And here we are 7 years later, where the use of CRISPR-Cas9 to generate cellular models or animal models of disease, to help us identify targets, to help us identify compounds, to translate the activity of those compounds towards the clinic, is probably impacting pretty much every drug discovery project that we run. And that's over 6 or 7 years. And that is truly transformational. And that will help us identify better medicines. It will help us accelerate the delivery of those medicines to the clinic. And that's before we even begin to consider the potential of therapeutic genome editing, which Jennifer alluded to, and we'll hear a lot more about this afternoon. And the ability to take CRISPR medicines that will actually, and here's a really, really interesting thought, CRISPR medicines that will actually cure disease. They won't treat disease, they will actually cure disease, restore patient health. That's not something that many medicines do, cure disease. And that's what we face as we go forward. And that brings major challenges for us as scientists, as an industry, us as society. The purpose of this session is to move away from what I know. So I'm now going to talk about something I know absolutely nothing about, which those of me who know me at AstraZeneca would say I do every day of the week. And we're going to talk about the applications of CRISPR-Cas9 in the agriculture industry, and how CRISPR-Cas9 may impact on our ability to improve the health of animals, maybe to improve food yield from animals. And an interesting way of viewing this is we often view what can CRISPR-Cas9 do to improve human well-being through editing of animals, through editing of plants. Interesting snippet just now talking to Helen. Maybe we should view it from the other perspective. What could CRISPR-Cas9 to do to improve the health and well-being of the animal, which in turn will help improve the health and well-being of mankind?